polypeptide can be very interesting in many applications. Of course, we like also polysaccharide. Elder has been showing nice example of polysaccharide that are very important and very interesting for nanomedicine. And we want to basically develop system that we can predict the way they will assemble and the way they will form different particles, I would say, of different shape. And among the shape, we have been very much interested over the last years. You have vesicles, namely polymersomes, but also spherical micelles or disc-like structure in some cases. We do control the self-assembly process using mostly microfluidic, and now we do, let's say, I think 80% of the system that we do design are made by microfluidic with all the physical chemistry characterization that you need to understand that. And of course, we want to design system eventually that can be used for biomedical application. And you know what we have in mind is really to use building blocks and develop chemistry so that the system in one step from the molecular coding basically would control both self-assembly and biofunctionality. So among the building blocks I mentioned already, we like very much polypeptide. And we've shown over the years that indeed to develop stimuli responsive nanoparticles for drug delivery. First, we demonstrate that vesicles made of polypeptide can be pH responsive and swell and shrink depending on a particular pH. But also, we developed more recently glycopolypeptide vesicle a bit more than 10 years ago that we try to transfer as much as possible to clinic. But today, I wanted to focus more about a new class of polypeptide that we have been interested in over the last years that are not really synthetic polypeptide, but bioengineered polypeptide. These are, we call the system protein-like polypeptides or protein-like omphophile. And the system allow to form nanoparticles with solvent-free approaches in a reversible manner, especially using thermoresponsiveness of this particular class of proteins, basically, with very high precision in terms of chemistry, but also in terms of reproducibility of the self-assembly properties that you can get with this system. So what kind of polypeptide or what kind of protein we focused on? These proteins are named elastin-like polypeptides. They are made recombinantly, meaning that we don't use synthetic methods, classical polymer synthetic methods to make them, but we use bioengineering. And this elastin are just a repetition of a pentapeptide sequence, which is valine, proline, glycine, an amino acid that we can change here at glycine, so VPG-XG, as many times as you want. And basically by changing the length of the sequence here and the amino acid that you have here, more hydrophilic or more hydrophobic, you will end up with a protein which has LCST character, eventually a UCST in some cases, that you can tune by just changing this amino acid, basically, as you can see here. So this protein just basically behaves as polynipam is behaving. So this is an interesting characteristics, and we have been using that some years ago to make micelles with the system by playing with, by designing copolymers with protein with different sequences. But I want to focus today on how this protein are viewed nowadays by the community. And basically these ELPs are now really considered as a model, as unique model of IDPs. IDPs are intrinsically disordered proteins, which are, you know, known or understood progressively to be very important in the cells. So it's not, they are not only there to make, you know, the environment, the cellular environment crowded, but they are also responsible of confinement of reactive species in particular conditions and induce particular cellular reactions in particular conditions. So these proteins are completely disordered compared to proteins that are highly structured. And this disordering is probably responsible of the LCST character of the polymer. The first question that we ask ourselves, I mean, we are polymer scientists, right? And we are not protein engineering guys. And we just ask ourselves, are these ELPs really behaving as polymers? And to understand this question, in collaboration with Toth Shikori, we designed a series of polymers, especially copolymers, but if we play below the LCST of the two blocks, basically, this system should behave as a homopolymer. And we just measure by small neutral scattering experiments the radius of gyration of a series of polymers of different molecular weight. And if you just plot the radius of gyration as a function of molecular weight, you end up with this linear relation here, and with a scaling law that is very close to the scaling law that we would expect for a polymer in theta condition, which is what you would expect for polymer with an LCST character. So this is a protein on one side with a perfectly monodispersed character, with a perfect sequence of your polymer backbone, but that behaves as a synthetic polymers, as poly-9' would behave basically in solution with a Gaussian coil conformation. So with that in mind, of course, we wanted to design, a larger series of proteins, of polymers. But if you want to make another polymer, in the polymer scientist background, you just change if you just want to change the molar mass, for example, you change the monomer to initiator ratio. Very easy. And after a few days, if you use a simple method, you would end up with another polymer. If you want to design a protein with another mass, you need to clone a new bacteria and use molecular biology, and it will take a few months to do it. So to avoid that, to make new proteins with a shorter time, basically, we wanted to develop a hybrid synthetic approach where we mix protein engineering plus post-modification, chemical post-modification. And so to do so, on any protein, you can modify them on the N of C-terminus, or you can modify them on particular amino acid laterally. And this is what we choose. And we choose to use a particular amino acid, which is methionine. And we collaborate with Team Deming to do so. We develop this methionine alkylation or thioalkylation reaction, which is very specific towards amine or carboxylic group that allow this modification. So now what we did is that by thioalkylation, you can take a single polymer, a single protein, and modify the protein with different functional groups. After this modification, of course, you end up with a new polymer with new properties. And if you look at, for example, just LCST character of the system, you will see that after thioalkylation, so first you see that you generate a charge during this chemical process. You will just increase the hydrophilicity of your polymer. You will maintain the perfect monodispersed character of the protein. If you look at the mass spec, you have one single peak. And if you look now at the effect on the LCST, what you see is that from the pure methionine, if you alkylate with a methyl group, you will lose the LCST, and you can eventually recover the LCST if you modify it with a benzyl group here that will bring a more hydrophobic character to the modification. You can play also with the same alkylation, but you can play now with the counter ion, which is kind of very obvious. And if you use different counter ions, iodine or fluorinated one that are more hydrophobic, you will either lose the LCST or you recover the LCST just with the change in counter ion into your system. So it's another way to tune it and to play with that. Another trigger you can play with is the oxidation of this methionine. And actually, in your body, methionine are there to be oxidated, and they basically take up from the body the reactive oxygen species that can damage your tissue. And when doing so, if you oxidize into sulfoxide, or twice, I would say, with making the sulfone, at the end, you will see that you will end up again with a polymer perfectly monodispersed, but that goes from a thioether with an LCST around 25 degrees to a sulfoxide with much higher LCST, down to a sulfone, which higher but lower compared to sulfoxide LCST character, because sulfone are less hydrophilic than sulfoxide, basically. So you can really tune this particular character. And of course, it's just LCST modification, right? And we have been doing a lot of understanding and scaling laws of the system. But now if you play with more, let's say, functional group, and use, for example, this particular alkyne that you can generate here, you can then do further conjugation. And we have been conjugated many different systems, such as drugs or oligomers, for example, of saccharide. And here I just want to show you an example on monosaccharide, disaccharide that we can use for conjugation. And this will give us access now to a new family of polymers that are kind of mimic of glycoproteins, right? Because you have a protein scaffold that you can glycosylate laterally with this chemical modification. So what we did is that we conjugated the system with two different sugars that are lactose or glucose or galactose, sorry. Galactose is particularly interesting because it's known to recognize with particular lectins, such as LCA120, and glucose does not recognize this lectin. What you can see here is just by light scattering, following the correlogram of the different system, you can see here the LCA120, which is very short time, so it's small molecules. And if you add into your system, you see the ALP galactose and ALP glucose alone. You have this correlogram here in blue and green. And now if you mix together ALP glucose, you see that you have no modification of the correlogram of the ALP glucose. But if you mix now ALP glucose and LCA120, you see that you have formation of aggregates. And the more LCA120 you add, basically the more aggregate you observe into your system. So it's a kind of dose-dependent aggregation of your system. So it's a way, I mean, using light scattering is a way to follow this interaction at kind of a molecular level. Of course, you can see macroscopically what happened is that if you indeed incubate ALP galactose with LCA120, you see this precipitation here, and you have no interaction, no precipitation if you have ALP glucose into your solution. And after redispersion of your pellet, as you have here you have no pellet, so no fluorescence. If we label now the lectin with a fluorescent dye, with FITC, and the ALP galactose, you see a lot of lectin that is redispersed from this pellet. If you look at the confocal microscopy, you see exactly the same information, but now it's in serum, so it's a more complex medium with a lot of other proteins that can be potentially competitive to the binding of your glycoprotein with galactose to LCA120. You see the initial state. When you add your glycoprotein, you see a lot of aggregation of your lectin, and this is here what you have when you add your ALP with glucose. You see no aggregation of your system. So this result shows that you can indeed selectively bind and sort lectin sugar, specific lectin due to this lectin sugar interaction, which is multivalent. I didn't mention that, but it's very important to have this multivalent presentation of sugar into a single backbone. And what is interesting here is that we use the LCST character that we still maintain onto the system to have a very strong palette that really allow efficient sorting of the lectin, including in a complex media. So again, you can take advantage not only of the binding interaction, but also to the LCST character of the system that allows very strong recovery of the protein of interest. So we wanted to go a bit further somehow in trying to mimic particular biological biomicromolecules that are very important. Glycoprotein is one example I'll just show you. But we wanted to go further and use this post-modification of ELPs to now mimic lipoprotein. And why doing so? Because, you know, protein lipidation is very important in living system and responsible, for example, of membrane trafficking. And it's a way protein are using to anchor into the membrane and to induce some particular signal or eventually for apoptosis. So how we can do that in synthetic system or in biomimetic system? The first way that has been done, or the first approach that has been developed, is based on post-translational modification. again, based on the protein engineering and enzymatic modification, basically, of the protein of interest. This has been done mostly by the group of Shilkoli. We wanted to develop a more, let's say, simple approach, again, using this combination of protein engineering and post-chemical modification, this time, using just chemistry. And so with a very similar backbone as I've shown before, we just use, again, this alkane group to modify it with now lipid tail or alkyl chain, more simply, that have been modified with the acyl group. And after a click reaction, we just hear kind of mimicking of an oleic acid system, the C18 chain. We could indeed verify by NMR that we have the perfect quantitative modification, again allowing to have polymers with a molar mass that are really perfect. You can control that by the integration of this proton or triacyl group compared to this proton here, or the 3 proton here that are on the protein scaffold, and you can see that you have a perfect correlation between these two numbers. And now we end up with an amphiphilic structure because we have a protein scaffold, which is the ELP, again, thermoresponsive, but with this hydrophobic alkyl chain. And if you just make a film and rehydrate, as you would do for the preparation of vesicles or liposomes, basically, if you first hydrate on quite large glass beads, you can observe here by confocal microscopy the formation of vesicle. If you zoom in, you can indeed observe giant vesicles with a membrane that you can characterize this way. Of course, they are micron size, but if you want to form nano-sized vesicles, you just need to hydrate the system on smaller beads here, 0.5 nanomillimeter glass beads. You observe here by now electron microscopy the formation of vesicles, and by light scattering, you see that the average numbers are very consistent. But we wanted to understand a bit more what is the structure of the system. So of course, to do that, we wanted to, or at least we wanted to have a deeper information about the thickness of the membrane, which is from a TEM experiment around 7 nanometer. And we end up with a model. So the model would be that, of course, we would expect the formation of a bilayer due to the alkyl chain forming the membrane, and the ELP would form the hydrophilic interface in your system. But that means, because lipids are laterally graphed onto the chain, that we would form loops here of ELP at the interface between this bilayer of lipids. And let's say the supposed model would be something like that. We can try to correlate this schematic model with numbers. First, I mean, we know that the thickness of pure PC, so basically a bilayer with C18 chain, is 2.7 nanometer, which would be this number. And now we have this loop of ELP in between two alkyl chain that we can calculate, or can estimate the radius of gyration of the system from the scaling law I presented just earlier in my presentation. So from the mass that we have in between two ELP, two alkyl chains, we can calculate this radius of gyration, which is a 1.9 nanometer for a linear chain. But we know that the radius of gyration of a loop compared to linear chain is about 1 over the square root of 2 from the linear one, which would give us this number. And so the size, this dimension here, would be twice this number, 2.5 nanometer. So in total, from this very schematic representation or schematic model, we would end up with this total bilayer thickness, which is about 7.8 nanometer, which is kind of good agreement compared to the one that we observe, making this schematic model reasonable, I would say, if not in good agreement with the numbers that we have. So now we have a vesicle, we have ELP at the interface, and this ELP potentially still have a LCST character. And we could verify that by just changing temperature. And if we just run DLS again, changing temperature, what you can first follow is the total intensity from a system, the derived contract, basically, which is the number of photons on your system. And what you observe is that if you go from 25 to 45, you see an increase of the scatter intensity, and you see at the same time, and this is perfectly reversible if you cool down. You see it's perfect reproducibility of your system. At the same time, if you go from low temperature to high temperature, you see a decrease of the size of your system. And so these two observations are in good agreement with the fact that you contract or collapse your ELP chain to the interface of your vesicle, making the structure denser. And because the structure is denser, the scattering is higher. And so now you come up with this kind of model where you have a vesicle at low temperature with hydrated ELPs that, when heating your system, collapse onto the surface and basically as a consequence of the dehydration of the ELP that follow a local ACST at the interface. So this is what you have at 37 degrees. And if you cool down, you see that you have a more, let's say, hydrated function. And so now what we did is that we did encapsulate another drug, which is just fluorescein, that is fluorescent, into this vesicle. So you see that with or without fluorescence, we have about the same size of vesicle. And now we were interested in following the fluorescence into the vesicle. And so we did encapsulate the fluorescein at high temperature, 37 degrees. We have a kind of low level of fluorescence. And if you cool down the system, you see that we rehydrate the ELP. We have a swollen structure, basically, of the vesicle. And as a consequence, you see a release of the fluorescein at this particular temperature here, which is around 37, basically, where you have the tunnel. And this is a function of time. So it's kind of really very precise on-off encapsulation release profile onto the system, again, due to these unique properties of elastin-like polypeptides. Let me check if I have some time. I don't have so much time, so I will go very, very fast in that. But this LCST character of these ELPs, as I said, are mimicking IDPs, intrinsically disordered proteins. And we wanted to use them in a very similar context, meaning mimicking somehow intracellular organelles that we may find in cells. And to address that, again, this was very strongly inspired from the work of Chilcotti, who demonstrated that the formation of coarcevates, not intracellularly, but in droplets, would prevent, or after interaction with particular RNA, would prevent the interaction with the protein machinery for the production of fluorescence protein, of GFP, basically, in that system. So here they use the coarcevation not to induce particular reaction, but to prevent particular reaction due to the protection of the enzymatic activity due to coarcevation. So we wanted to demonstrate that we could design a system that is not as complex as in cell, but that go through a close comparison to what cellular complexity is. To do so, we made droplets, not yet vesicles, that would contain a mixture of dextran and PEG, which is known to induce a phase separation based on temperature. So here is what you have at this is the phase separation that we would have with this mixture at 37 or at 45 degrees here. So high temperature, and this is the low temperature limit. So basically, if we are here and if we cool down, we will go now on one part or the other part of the phase diagram. So when cooling down here, you see the phase separation occurring. And what you observe with the ELP labeled in blue is that the ELP start to be homogeneously with a homogeneous distribution at the initial state, and they will migrate to the PEG phase after cooling down to the system. So just briefly, we made an even more complex mixture by mixing a dextran, PEG, and two proteins, BSA, as the neutral proteins that will confine mostly, and it's known, into the dextran phase, and or ELP that after heating or cooling will form coacervates and will go at a particular location. And what you observe is that if you mix everything, we start at 50 degrees. If you cool down, so after phase separation, at some point, everything is mixed together. And if you heat up your system, what you would observe is that we would have a perfect distribution of the ELP here at the interface between PEG and dextran, forming these blue droplets here that are coacervates. And BSA will migrate into the dextran phase, again, see with this co-localization of dyes in an almost perfect manner. So this allows distribution of different proteins, a particular region of a complex, let's say, lumen that will mimic a cell. And again, what is very interesting is that we can use the LC of ELPs to induce homogeneous distribution or particular localization at interfaces. And of course, what we want to do now is to use this process to make interfaces and to use these interfaces to do chemical reactions or biochemical reactions, especially bioenzymatic reactions that will be controlled in space and time due to this particular confinement reaction. So I just want to finish up, and I will be brief with concluding remarks. I focus mostly on the particular LCST character of these elastin-like polypeptides, which are polymers, but also proteins, and that combine unique properties of both elements, I would say. This really offers new opportunities in design of functional materials, but also I believe new opportunities for the understanding of LCST or UCST character and phase diagram of theory, because the systems are really perfectly monodispersed, and you can really control every single charge or every single element that you put into your system. I want, of course, to thank all the students and collaborators who have been involved in that work. I'm mostly focused on the work of Vusala, Monica, and Rosine here in my presentation. I want to thank all the collaboration, and of course, I want to thank all my group and you for your attention.